for me to be able to come to this whole workshop and see the issues that you're dealing with on a daily basis. And I want to tell you today about some of the work that we do at the Center for Plant Conservation. Your workshop has really been focused on the importance of genetics for helping us determine research questions, the answers to research questions. But sometimes you do not have genetic information. You don't have the study there in front of you. And some of the work that I'm going to be presenting to you is broad best practices that can help you maximize genetic diversity without having discrete genetic information. So a little bit of background. Next slide, please. The Center for Plant Conservation now has 68 conservation partners, now from 4 different countries. And we hold 2,100 globally rare and 70% of the federally listed plant species in the United States. We advance science-based best practices, and those are the practices that I'll be talking to you today about. And we also advocate for plants and their benefits to humankind. Next slide, please. So, very important point is that there's a lot of experience within the Center for Plant Conservation Network. Collectively, we've done over 150 plant reintroductions. We've done some publications on that work. Also, that can be a help to you. And today I'll be telling you about some of those that have genetic implications to them. Next, please. The previous slide had some photos of plants that colleagues and I reintroduced in South Florida, so closer to tropical area like you experience in Hawaii. But at San Diego Zoo Wildlife Alliance, we're also doing reintroductions. We've done one of Dudleya brevifolia, and we're also working with Torrey pine. Next, please. And these best practices have been summarized in these publications over the last 20 years. And within these publications, you could find guidelines for reintroduction planning, for managing conservation collections, and for making collections. Knowing that the world is not all easily going to a bookstore, and carrying 4 books into the field with you is not the easiest thing to do. We have gone to a digital platform. Next slide, please. So we have actually a printed version of a collation of these best practices. And next slide, please. We have an online resource for best practices. These are under the title of the Center for Plant Conservation, but there are a lot of conservation officers who have contributed to these guidelines, and we've tried to make them as inclusive as possible. Here are some of their beautiful faces. Next slide, please. There's some people missing though, so I've got to get better slides. So I wanted to give you an example of how these guidelines help us. And in this case, we've got an opportunity to look at what you can do if you don't know anything about genetics, and look at what the genetic information has told us too. In San Diego County, there is a vernal pool species called Acanthamintha ilicifolia. It it's an annual that has a very ephemeral boom bust cycle. And so it was federally listed because when there is drought, there are very few individuals that make it above ground, and it's very difficult to track. We are now, gathering decades of information about the species, and that is enabling us to have a better sense of what's happening with it. But next, please. And next. When we're making conservation collections, the first dictum is to do no harm to the population. Next, please. And so in order to do no harm, we have done modeling to determine how much of a seed crop you could extract from a population without doing harm. And the research we've done to date suggests that you can do at least 10% without doing harm. We have current studies underway to check on that with species such as this annual that I'm talking about today, which has these huge boom bust cycles. It may be that in good years and bad years, you can adjust that percentage, but please stay tuned for that information. Next, please. You strive to maximize the genetic diversity in the collection by spatially collecting plants that are spatially dispersed and have different morphologies and maybe in slightly different microsites, just to try to capture as much as you can. Next, please. And from at least 50 mother plants in a collection, or if there are not 50 plants that are reproducing, to collect from all of those plants. Next. From at least 5 populations across the distribution of the species. And next, capture, as I mentioned, the diversity across space, time, appearance, and ecology. Sometimes that means that you may have to make collections early in the season, late in the season, 1 year, and across years to capture full diversity. Next, please. And as you're making these collections, to not group the maternal lines into a single package, to keep them separate. There are 2 important things that happen with that. One is that you get a sense of the total number of maternal lines that you've captured in the population, but you also get a sense of perhaps what 10% of each of those moms' seed productions for the year has been, and there's going to be variation in that. Next. We also recommend that with each seed collection that if the population can withstand it, taking a voucher specimen of it, and if a voucher has not been collected from that population in the last 5 years, it's a good idea to take a voucher specimen of it. But to also capture leaves and place them on silica for potential genetic studies. Next slide, please. And realizing that as you're collecting, this is not a stamp collection, but it is a way to support the species survival in the wild. So using that collection is really a big component as well. Next slide, please. So I often use this in courses with students. These black diamonds and those blue dots that are on this screen are populations of San Diego thorn mint. And I asked the students, if you were going to capture genetic diversity of this species, and you only had the time and the resources to capture 5 populations, where would you collect? And the students will pick something that's way up here in the north and way down here in the south, and then they'll take something over in the east, and they'll probably take something over here in the west, and then something in the middle. Because they're looking for spatial distance as a way of looking at diversity. If the people know exactly the habitats that are represented on this site, then you get a more refined assessment of where you would go and do collections. This area, for example, over in the east is up in the mountains. It's higher elevation than this one that is over on a mesa over in the western part of the county. And so not knowing genetic information, that's kind of an approach that would be advised. But if you do know genetic information, the next slide, please, you can see that it gives you a similar pattern. The circles of different colors represent different genetic groupings as assessed by actually a RADseq analysis. So again, way north, way east, something in the west, something in the south, and something in the middle. Next slide, please. But those genetic studies, if you've got the resources, can really give you more information about where you're seeing populations that might surprise you as being similar, because not always is it going to be geography that determines similarity. And I'll show you an example of that later. Next slide, please. This actually has been published too, but I can't remember whether I gave you that publication. Just contact me if you're interested. So we've taken these best practices to a digital platform online at saveplants.org. And one of the ways that you can use genetic information is if you're planning a reintroduction. And so I'm going to show you some examples of how that has been used. Next slide, please. So again, in making a new population of a plant or bolstering an existing one, genetic diversity is really critical. And by maintaining maternal lines separately, you can maximize diversity consciously. And I'll show you an example of that. You have to have adequate numbers of plants or seeds to begin the reintroduction, because if you have a small number of founders, which is unfortunately a lot of what our problem is with plant reintroduction, that we just can't get enough propagules to begin with to flood a new area. Select the source material with similar ecology, climatic, and environmental conditions to the recipient site. And hopefully that is matching a genetic with a location situation. And determine whether to use propagules from a single population or a mixed population. Next slide, please. So an example of why maintaining family lines evenly and knowing who a seed's mother is illustrated here. When you're growing plants in a greenhouse, it's really common that one mother, the seeds of one mother, or even the cuttings from one mother, are going to grow more vigorously than another one. And if you don't really keep a conscious tally, and actually a numeric tally of where the seeds come from, it's really easy to pick the biggest and the best. I mean, that's what we have traditionally done in horticulture. And I know that in Hawaii, you guys have this down. You know, you are our showcase and our are leaders in this idea. You absolutely keep track of who the mothers are. And you do that for many reasons, including sometimes you're discovering new species that are arising within populations, I know. So what the idea is when you're maintaining these even family lines, next please, is that you want to avoid artificial selection and allow natural selection to take its course. It's a possibility that natural selection will select for all of these or most of these large individuals. But it's important to give natural selection a chance to do that rather than you doing that. Next, please. So because genetic studies are expensive, one of the pieces of advice that we give in our best practices is this series of questions. When are genetic studies needed? And this is our advice. And actually, this comes from the Wise K. Havens. Within populations, if a population has fewer than 50 individuals that are flowering and setting fruit, if the species is clonal, if it has little or no viable seed being set, or if there are potential taxonomic concerns, some ambiguity, you would want to have genetic studies to help you understand this. And across the species range, if the species is declining and little is known about the biology or life history of the species, if the species has highly fragmented and isolated populations, if it looks different in different locations, and if one or more populations of the species has distinct ecology from the majority of the populations. Genetic studies could help you understand what's going on with any of these. Next. So I'd like to just review with you some of the terminology that's used within the US and in the world when we're talking about reintroductions, so that you understand something that you may be reading about this. The terminology that's used depends on the source population and where the recipient population is located. And so next, please. You're starting out with a source population. Usually you take seeds or cuttings from that source population, you propagate them at a botanical garden to bring up the numbers. And then if you put those same genotypes back into the source population, that's known as a reinforcement, an augmentation, or an enhancement. Next, please. And this is, first of all, going to increase the population size. That's a no-brainer. It can increase the genetic diversity, especially if the seeds or cuttings that are taken to the botanic garden are there in a different time frame than when you're putting these back out into the wild. So for example, if you collected the seeds 5 or 10 years ago and you're putting them back, they're going to be most likely genetically different than what is currently in the population. And we can talk about the good things and bad things about that. And this technique can also increase specific demographic groups. I've experienced this with a long-lived palm in Florida. We propagated the palms in our botanic garden. They became 6-foot high plants that we put back out into the field. And that particular age and structure class within this population was absent. And so the reintroduction, or actually it was an augmentation, helped the age distribution and spatial structural distribution in the population. Next. So you can also have a source population, botanic garden, and an unoccupied recipient site. And next, please. If this site was previously had the plants there, it had the species there, but is currently unoccupied, then this would be known as a reintroduction. And next, please. Next, please. And this would be within the historic range of the species. In Europe, they tend to call these things also conservation translocations. We don't use that word translocation much in the US because in the plant world, a translocation means that you have dug up the plants from the wild and you've moved them to another location. And generally we frown on that unless the initial population is going to be destroyed for some reason, which sometimes happens in development. Next, please. And next. And if you have a situation where your unoccupied recipient site is appropriate, but never had the species growing there in the past, this would be called an introduction or a conservation introduction. Next, please. And next. And this terminology is used to describe something outside of the historic range. So where you have not seen the species in the past and outside of the range. Sometimes that is also called an assisted migration, a managed relocation, or assisted colonization is the other term that is sometimes used. Next, please. So before doing a reintroduction, there are genetic questions to consider. It's very traditional to use a single source of material when planning a reintroduction, but it's becoming more common to also use mixed sources. And that's, I think, a consequence of our rapidly changing world and the low numbers of propagules that are available to use as founders. If you can mix these sources if the wild population has inbreeding, declines in vigor, or low reproduction. And the concern about mixing sources has been outbreeding depression, but outbreeding depression is unlikely if the existing populations are small, have no chromosomal differences, no distinct ecological differences, and have been separated less than 500 years. And I think I gave you the Frankum paper to look at for those recommendations. Next, please. So here's an example of a series of reintroductions that my staff and I did at Fairchild Tropical Botanic Garden with the federally endangered species Jackmania reclinata. And in this case, we were taking action somewhat simultaneously. We had the luxury of doing genetic studies ahead of time. And in these days, the type of genetics we did was AFLP. And what we discovered was that only one of the 7 populations that we studied was so different genetically that we would not mix them. So these other ones, we felt that it was safe to mix, but not with this, with letter G there that is being denoted. And Gary, if you could skip 2 slides, please. So we identified the existing populations, we mapped them, we knew how many individuals were at these populations. And I'm going to be telling you about a series of experiments that were done at South Beach and Crandon, the kind of the middle place in the distribution, or the northern middle and the southern distribution. And the genetic work told us that there weren't genetic differences between these two populations, which really puzzled me because there's about 90 kilometers difference. But then I experienced, Gary, next please, my first hurricane. And hurricanes really can span and lift up seeds and carry them 90 kilometers, no problem. And so that may explain some of the mixing that we were seeing in this population. Now, if you could go backwards, please, Gary. So simultaneously, we were doing experiments on breeding history on this species. This is the work of Elena Pinto-Torres, and she did 5 pollination treatments using the South Beach and the Crandon Park populations. Those were at the time the 2 largest populations of this species along this eastern coast of Florida. And she had a control, she had self, siblings were within a meter, far neighbor was over 20 meters away, and between site crosses were between Crandon Park and South Beach, so across this 90 kilometer stretch. And if you can skip ahead to now. And what we found was so Elena did her study, she found out that there was a mixed mating system and that there were some fitness advantages of this cross-site pollination. And when we put those individuals out into wild sites, what we found was the same thing, that the survival of the individuals that were from mixed sources was greatest in comparison to the self populations. Next, please. So that's an example of one way that one could use genetic work to inform what you're doing in a reintroduction. And of course, genetic work since those days is getting much more sophisticated with genomic research. This is an example from Van Rosen et al. In 2020. And they were actually interested to see whether when you translocate or reintroduce a population, whether or not you are conserving the genetic diversity. And so they had source material from 2 largest populations of Arnica montana. They grew plants in their nursery, so they had 700 10-week-old plants from 2 mixed sources and introduced into a 3rd recipient site. So they augmented 2 sites and they introduced into a 3rd recipient site. They fenced the areas, they mowed the other vegetation, so they reduced competition for the plants. And what they were really asking was, is the translocation conserving the genetic diversity of the species? And next, please. They did have sexual recruitment after 2 years, and the translocated populations exhibited high contemporary pollen flow, substantial admixture, and low inbreeding in the F1 offspring. There was no genetic differentiation between the generations within and between the translocated populations, and no evidence of outbreeding depression. And so their conclusion was mixing the source populations resulted in high genetic diversity. They realized that there is some evidence from Frankham 2015 that if you're going to see evidence of outbreeding depression, that you need to look at the F2 and F3 generations. And so their plans are to continue doing that in the future. And next, please. Another example of how genetics has been used in reintroductions is this one from Monks et al., which was published 2021. Their source material was single wild populations of 2 subspecies of Lambertia orbifolia. And they did genetic studies of the translocated populations. Again, they're using, these are conservation introductions, but their terminology, translocation, outside of the range of the species. And what they discovered was that the genetic work also elucidated the mating systems. So 2 subspecies of Lambertia orbifolia. Next slide, please. And over on the left is the population, or is Lambertia orbifolia. It's a little complicated, but what they started out with was a distribution of Lambertia orbifolia that they thought was a single population. And now with further genetic work, they see more differentiation as the populations have been fragmented. But you can see the 2 bars that are, or the 2 columns that are off on the far left have all of the colors that the 3 middle groups have, which means that there's intermixing of those populations going on. The subspecies South River Plains used the BNUP, yes, source population, and you'll notice that the BNUP, again on the far left of the screen of the orange and green, is showing a mix of both BNUP and the Dennis Road genotypes. So this is another opportunity for using genetics with reintroduction to understand how the gene flow is happening in these populations. Next slide, please. So in summary, genetic work, genetic considerations are important as you're making collections, when you're maintaining material in the greenhouse, and when you're using the material for conservation translocations. Next. I just wanted to quickly show you an example of a hybrid situation. This was one that I did in the past where we also had some genetic work. And to show you that when we started out, if you look on the top right, Persia subintegra was described morphologically as having these spatulate, non-lobed leaves. The species that's on the far right, Persia stansburyana, has lobed leaves, 5 lobes on its leaves. And there's other differences across these species, but it's clearest to see in this leaf lobing trait. We began to notice that there were these plants that had leaves that had an intermediate number of lobing. And when we did genetic tests of this, we discovered that there was a big hybrid swarm that was integrating with Persia subintegra, the federally listed species. Next slide, please. And we saw that this in the evidence from this principled coordinate analysis as well. And noticed also that only a single one of the populations that was named Persia subintegra really stood out from this hybrid swarm completely. And actually, historically, it had been named a different taxon, so stay tuned on that. But I just wanted to show you this information that also genetic work can help you understand the morphological differences that you're seeing in the population. And also, if you are making collections and noting what the morphology is of the taxon that you're collecting from, you can also use that information for your reintroductions. Thanks so much, and it's been a pleasure talking to you.